polymerase chain reaction, there are two concepts that we need to highlight. Those are the primers. Let me put my notes here, my pen. So those are the primers and the probes. Two concepts that are needed to be part of the PCR experiments. And there is something common between primers and probes. They are both oligonucleotides. Oligonucleotides means they are both some short sequences of DNA or RNA. That's the commonality between both of them. Usually these are synthesized. So you make up some kind of sequence, one to help and to be used as a primer to help in certain task, and the other one to serve another goal. But both they are short sequences of DNA or RNA. Now, when it comes to PCR primer, they are used to anneal, as you see here. The primers are used to anneal, that is bind to the complementary sequence in the DNA template that we see here. Okay, so they need to bind there, and they provide this starting point for the DNA polymerase, as we have seen earlier in that video. So with regard to PCR probe, it is designed to detect the presence of amplified DNA. Okay. So we have a target sequence that is, now we are not dealing with a primer that's going to be then part to start initiate the DNA polymerase side and then have synthesis. In this case, we have the as you can see, a probe that can bind to a target sequence. So it is designed to detect the presence of an amplified DNA. That amplified DNA can be one that is part of the PCR process, takes place during the PCR or after the PCR. So there are these probes usually they are fluorescent probes. You see there's fluorescence, so some light is to be or some emission is going to take place, and there's a quencher, which we can think of dark fluorescence that kind of balance absorbs the fluorescence under certain conditions. We're going to come to this in a later slide. So probes, in this case, the fluorescent probes, they have this labeled dye and a quencher, as we can see here, in this case, F and Q. And when the probe is intact, the proximity of the quencher prevents the fluorescence. So during the PCR process, when the probe binds to its target, as you can see here, it binds to its target. Okay, we have different steps. When this happens, when the probe binds to its target and it's degraded by the DNA, that is when the quencher gets separated. That is when the quencher gets separated. So this is where PCR probes come into play. They are more of a tools for detecting and sometimes quantifying the amplified product. They are part of what we'll see in the coming slide that is called real-time PCR applications. So primers are essential for initiating the DNA amplification. So that is more focused on the initiation of that DNA synthesis, DNA amplification process in PCR. Probes, on the other hand, are tools to detect and quantify that amplified product, So they have two different roles in this process of PCR and the ways that they can be used. So let me take you here through this figure a little bit with some discussions, because there are certainly a couple of terms here. We understand there is a probe, there is a fluorescence, and there is a quencher, and that is what they can refer to as a fluorescence dye or reporter dye and a quencher dye. The fluorescence or the reporter dye is usually located at the 5' end. And this is the quencher usually at the 3, is situated at the 3' end. So reporter dye is playing a role in emitting fluorescence when it's excited. Now, when there's no excitation when it's intact, in the case of intact probe, the fluorescence is quenched by the proximity of the second dye. So this keeps some kind of control when it's compared at different stages of the, as you see here, there's the annealing and trying to have a bind and see what's going to be the outcome in terms of quantification. The combination of these two dyes, the reporter and the quencher dye, in a single probe gives more specific detection method that is used in real-time PCR. So yeah, primer is part of DNA synthesis, the amplification step. Probes, they are designed specifically to see if there is going to be some binding and to the target sequence, and then that is, we are focusing on the amplified one, and kind of we designed this probe to see if that the presence and to measure the presence of certain gene, and eventually, so far now we understand there's always going to be some fluorescence somehow that will be released under different conditions. In this case, there is a need for another, let's call it dark fluorescence. You can maybe use this for the quencher. If this quencher, it's more of a chemistry term. So you can think of another type of, just think of it as a dark, this is like some light here, and this absorbs this unless there is an activity that's gonna release this and degrade the probe. So as part of the technology. Now, this is part of TACMAN assays that use these dual labeled probes. So they have the two types of dyes, and they are used in various applications such as gene expression analysis, I've just mentioned, trying to quantify genes, genotyping, and other applications that would require high sensitivity. So looking at the concept of primer, the primer in PCR should follow certain principles in terms of design. For example, in terms of length, they are between 18 to 24 base pairs. There is a melting point, as you remember, where it should be needed for the interactions or for the annealing to happen, and the different cooling and warming that is part of the PCR operation. So you can see here that GC content usually should be between 40 to 60%, meaning the guanine, cytosine, also some of the concepts that we covered earlier. There are different types of primers that are used for different purposes. For example, there's the gene-specific primer that we can see here. This is designed to bind specifically, that primer, to a unique region of known DNA. Okay, so we have specific interest, and you can imagine this. See, in this case, this is being used for the purpose of detecting specific genes if they are there, and so on and so forth. There are random primers, as you can. Sorry. So, okay. So random primers is another type of primer. They are, again, short oligonucleotides of random sequence. Something here, ends would refer to anything. It could be A, C, G, T. And those random sequences can anneal to different sites. This is used usually in cDNA, complementary DNA synthesis. In terms of application, there's standard oligo DT. This is what I'm going to put the treasure hunt question based on what you read here in front of you in the slides. This contains a series of what? Thymines, okay? Thymines or thymidine residues, what we can call. And you see this is trying to detect specific area in the mRNA. So I'm going to leave it to you to tell me what do we call this area from the previous lecture. And so there are different types of primers that are used for different purposes. Now, PCR, although it's a very powerful technology, as anything we have, you can imagine there are going to be always some kind of biases, or some issues that can take place in this process. So, okay, I have missed something very important because it relates actually to the biases, okay? Now, there are two concepts here, primers, dimers, and hairpin formations. Primer diamonds and hairpin formations, it's going to take us again to the bias, but quickly, primer dimers occur when two primers, they could be the same, or they are two different copies. They anneal to each other. And then this is going to cause an issue for the PCR amplification. This can lead to reducing the PCR product. So imagine there are two primers anneal to each other, and that is going to cause an issue. It may happen. There is a hairpin. It's a different thing. Similarly, it's when in the case of in context of PCR, when the primer itself might fold back. It might fold back on itself due to some internal complementary sequence, making the shape of a hairpin. And so these issues may happen, and therefore there are recommendations to use specific types of primers that are commercially available. But again, would get me to this discussion again, that there are some challenges in general that we can face in PCR. So we can face what is called chimeric molecules. Chimers form when two different DNA molecules are joined together during PCR. So if during the process of growing a DNA strand, detaches from the template and then reanneals. When the DNA strand detaches from the template and it and then reanneals to a different template, that certainly can cause an issue. Imagine this: we're working with one type of strand, we end up targeting that it's in the process, PCR in the process of looking and doing the synthesis between the primer and the template and so on and so forth. This happens. There are other types of issues that may happen: heteroduplex molecules, Taq polymerase errors. For example, Taq polymerase errors when the enzymes commonly used in PCR, these are the Taq polymerase, they lack proofreading activity. Something we discussed right in the first module is DNA polymerase, and that there is a need to go and do some proofreading because some mutations may happen. Taq polymerase lacks this ability or this needed mechanism, which means it can introduce certain errors as it synthesizes the complementary. It can the complementary DNA strand. Sequence artifacts due to PCR error and cloning. It's something wrong that goes during amplification or cloning. It's kind of an error that PCR cell might introduce a mutation while cloning, and so on and so forth. So different issues. This also can highlight of the the different types of biases, errors, and how they would actually eventually impact the read count, and trying to understand or quantify the specific gene of interest. So hopefully this can serve as a kind of reference. What I want to move to now is the applications of PCR, polymerase chain reactions, and how we can use them in different technologies. So let's watch this together and see what kind of applications we can use a PCR for. Now, before I jump to this and start playing this, I want you to share with me at least 2 ideas of what you think, based on our discussion so far, PCR can be used for in general. I guess I've mentioned a couple of ideas. It may seem a little bit, not clear, but I hope you get some idea that PCR can be used for different types of things as a technology. So I want you to imagine that. Now, it depends. There are different types of PCR, something that will be clarified as you watch this nice video, what kind of different PCR technologies are there and how they can be used differently. But as of now, go ahead and share with me at least 2 things you think we can use PCR for. Oops.
Hi there, I'm Jamie Dyer. PCR is a technique that makes lots of copies of a specific sequence. Hi there, I'm Jamie Dyer. PCR is a technique that lets you make lots of copies of a specific sequence of DNA. Now, if you don't know how PCR works, the rest of this video won't make sense. So go check out this video if you need a refresher. Okay, so you know how PCR works. RT-PCR is a really cool technique that actually, RT-PCR is 2 different techniques. The RT in RT-PCR stands for either reverse transcription PCR or real-time PCR, which are really different techniques. Sometimes scientists use the term qPCR to mean real-time PCR, but the naming convention is not standardized. So if you see the term RT-PCR, make sure you know whether they're talking about reverse transcription PCR or real-time PCR. And it's worth noting that these 2 techniques can be used independently on their own, or they can be combined. I'll tell you about that in a second. Reverse transcription PCR is PCR that uses RNA as a template. Now, for a normal PCR reaction, DNA polymerase makes copies from a DNA template, not RNA. And in fact, DNA polymerase can only copy DNA. It cannot use RNA as a template. But what if the thing you're trying to copy is RNA and not DNA? Like, what if you want to know whether someone is infected with the COVID-19 coronavirus? You can't do regular PCR to detect coronavirus DNA in a sample because the coronavirus doesn't have DNA. It stores its genetic information as RNA instead of DNA. So to do PCR using an RNA template, you first copy the RNA into DNA using an enzyme called reverse transcriptase. Remember, transcription is when RNA polymerase copies DNA to make an RNA molecule. Well, reverse transcriptase copies RNA to make a DNA molecule. That's why it's called reverse. Since reverse transcriptase used the RNA molecule as a template, the sequence of that new DNA molecule is complementary to the RNA. So we call it cDNA. Well, once you have cDNA, well, that's just regular DNA you can use as template for a regular PCR reaction. But since you started by copying RNA into DNA using reverse transcription, we call it reverse transcription PCR. Right, so to test if someone has the COVID-19 coronavirus, scientists used reverse transcription PCR because the coronavirus has RNA, not DNA, as its genetic material. But let's say you don't just want to know if the coronavirus is present, you want to know how much is present in the sample. That's where real-time PCR comes in. For example, in this paper, scientists wanted to better understand how the COVID-19 coronavirus damages the human body. Now, we know the COVID-19 coronavirus is a respiratory disease, right? It affects the lungs. But we also know that in some severe or fatal cases, we see damage from the virus in different body parts. So the scientists who published this paper, they wanted to know how many copies of the virus were found in other parts of the body and how that correlated with damage in those body parts. To study that, they did autopsies on 10 people within hours of them dying of COVID-19. They took samples from tissues all over their bodies, but they wanted to know not just whether those tissues had virus particles in them, but how much virus was present in each sample. To figure that out, they used real-time PCR. Remember that normal PCR is an exponential process. If you start with just one copy of the DNA template, you'll have 2 copies after the first cycle, 4 copies after the second cycle, and so on. Now, if you start with 10 copies of template, that's a sample that doesn't have very much virus in it. Then in principle, after 35 cycles, you'll end up with over 300 billion copies of the DNA. And if you start with 10,000 copies of template, after 35 cycles, you should end up with 300 trillion copies of template. So what?
So what I want you to notice here is the starting point of a sample. Okay, bear with me. The starting point. So this is the quantification that we're looking at. There's certainly this genetic material itself that holds certain quantities as a starting point of some specific mRNA, right? So this relates to the concept of cDNA, which is one very important treasure hunt question, and it's going to come perhaps in some review later on. What is cDNA? So think about it as you watch this in the lecture. And again, back to this, why do we see here this golden or yellowish line and the bluish line here, the blue line, at different positions? Because the starting point of the genetic material is already different. And as the quantification go ahead, we can see from the produced curves which genes or which material were there in higher quantities. And that's eventually going to help us in measuring gene expression. So I'm going to take you back a few seconds here so that you pay attention into how we can use real-time PCR to quantify the levels of the genetic material.
So not just whether those tissues had virus particles in them, but how much virus was present in each sample. To figure that out, they used real-time PCR. Remember that normal PCR is an exponential process. If you start with just one copy of the DNA template, you'll have 2 copies after the first cycle, 4 copies after the second cycle, and so on. Now, if you start with 10 copies of template, that's a sample that doesn't have very much virus in it. Then in principle, after 35 cycles, you'll end up with over 300 billion copies of the DNA. And if you start with 10,000 copies of template, after 35 cycles, you should end up with 300 trillion copies of template. So why can't you just look at the end of the PCR reaction and see which sample has more DNA in it? I mean, that's the sample that started with more template DNA, right? The problem is the PCR reaction runs out of nucleotides before it can make that much DNA. So what actually happens is something more like this. At the end, both samples have the same amount of DNA, but the sample with more starting template made more DNA faster. So what real-time PCR does is it monitors the amount of DNA in the PCR tube in real time after every cycle. By comparing the curves of the samples with the curve for a known amount of template DNA, you can know exactly how much DNA you started with. That's why real-time PCR is often called quantitative or qPCR. One quick note, you can't do real-time PCR using a standard PCR. How much DNA?
An additional note here is that this is also why in real-time PCR, it is typical to see curves that we deal with. Copy values, and usually we refer to CT values, which is going to see in the coming slides as well. So we have cycle number in real time, and for each cycle we know the copies.
Started with. That's why real-time PCR is often called quantitative or qPCR. One quick note, you can't do real-time PCR using a standard PCR thermocycler because there's no way to monitor how much DNA is present after each cycle. So to do real-time PCR, scientists use a fancy machine that can heat and cool like a normal PCR machine, but it can also measure the fluorescence of the PCR sample after each cycle. See, for real-time PCR, scientists add a dye to the PCR reaction that fluoresces in the presence of double-stranded DNA. More DNA means more fluorescence. By measuring the fluorescence after each cycle, the real-time PCR machine reports the amount of DNA product in real time during the PCR reaction. So remember the COVID-19 study? The scientists combined reverse transcription and real-time or qPCR to detect the amount of coronavirus in various tissues from the bodies of people who died from COVID-19. They found the highest levels of virus in the lungs, but they also found various levels of virus particles in tissues throughout the body, which can help us understand how the COVID-19 coronavirus attacks the human body. RT-PCR in both its meetings helped scientists answer some really interesting questions, including how the COVID-19 coronavirus kills people, which, if we understand it better, can help us save lives. Now, that's cool.
So nice. I hope you enjoyed watching this video. Nice. So here we get into, I guess there's a typo here, and what we're looking at is quantitative, right? Quantitative PCR or real-time PCR. And remember from that video we have watched, there are reverse transcriptase PCR, and there's RT. That is one RT, but can be confused with real-time PCR. That is used for one application. Here, real-time is used for another application, right? So hopefully this is something that can be used as a hint for your own answer on the treasure hunt question that we had just before we run the video. And this is the curve that we saw in terms of the relative fluorescence, going to give us access. Well, that was the cycle number and number of copies. And they also can look into the relative fluorescence in terms to indicate as well how much is there from that specific gene. Two types of methodologies or technologies, whatever you want to call them, is what's called that can be used, CyberGreen and TACMan. They are both used methods used for real-time quantitative PCR, but using different purposes. Okay, so quickly in terms of how they detect the target, the PCR in the CyberGreen part here, as the PCR amplifies DNA, more CyberGreen dyes bind to the newly synthesized double-stranded DNA. Okay, more dyes. It's based on these kind of green dyes. The fluorescence is then measured after each cycle. The fluorescence is used to be measured after each cycle. TACMan, in TACMan, the TACMan probe needs to the target sequence between the forward and the reverse, between the forward and the reverse primers. So we have reverse primer, forward primer, and here's the probe that we remember from the previous slide. And then there's a process of extension cleavage that's going to take part as the DNA polymerases extend the primers and synthesize the new DNA strand, which is this area. Okay, this is extension here. Okay. Oh, sorry. Yes, this is the extension here. This is that. eventually the extension. As the DNA polymerase extends, the primer synthesizes a new DNA strand. The exonuclease activity decreases the TACMAN probe, eventually separating. This is going to be decreted, releasing, separating the fluorescence dye from the quencher, and resulting, once they are, and again, the quencher is, as long as we keep the probe here, and the quencher is linked to it, and the fluorescence linked to it, the fluorescence is not released, light is not detected. And also as you said earlier, and to make it clear again, the quencher is trying to keep this high sensitivity, right? By not allowing this molecule, this fluorescence, to glow. Okay, it's keeping it under control for high sensitivity and accuracy of measurement. And then this is eventually can be measured. So two different strategies, both they use fluorescence. Here, one green fluorescence, kind of the traditional way as we have looked earlier. And here we look at things a bit differently with the probe design that we looked into earlier. So let's watch another video here for qPCR and learn more about the curves. Oops.
qPCR, quantitative PCR, or real-time PCR is an effective technique to quantitatively determine the presence of a target sequence or gene in a sample. Up to date, qPCR is the gold standard method to test patients for viral infections such as COVID-19.
Oops. Yep, I think there is some.
For that,
bear with me a second. I'm just trying to. I think we have two repeats here in the slides. Okay.
is an effective technique to quantitatively determine the presence of a target sequence or gene in a sample. Up to date, qPCR is the gold standard method to test patients for viral infections such as COVID-19.
And this is a very nice, interesting figure. We look into RNA, and again, what is cDNA? So we start with an RNA, reverse transcribe that to get the DNA, the one that we can go and process as part of the qPCR. And RNA is the material that really gives us the information of how much we have in terms of production for that gene. Remember, genes always, we have the same gene, but the process of transcription is going to give us the amount of RNA. So we cannot start with the DNA. We have to start with RNA. But again, we have to reverse transcribe that to get the complementary cDNA and then be able to work with that to do the synthesis and the different operations needed for PCR.
For that, a swab is taken which may contain viral material. The RNA is extracted and reverse transcribed into DNA. The DNA can then be analyzed with qPCR in a light cycler. In conventional PCR, a DNA sequence of interest is amplified. The presence or absence of that DNA sequence is detected in an endpoint analysis after reaction completion. During quantitative PCR or real-time PCR, the accumulation of the amplification product is visualized with fluorescent reporters, and the reaction progression is monitored after each cycle. In qPCR, the product amplification is therefore observed in a quantitative perspective. With each PCR cycle, the amount of PCR product doubles, which can be detected as an increase in fluorescence. This generates a classic qPCR curve. A defined signal intensity has to be exceeded until the product is abundant enough so that the fluorescence is detectable over background. The cycle number at which enough amplified product has been accumulated to exceed that threshold is referred to as the CT value.
I want you to pay careful attention for CT value specifically. CT value specifically. So I'm going to repeat this part. It is a single value that is used when a qPCR technology is used. Our aim is to get these values. As a bioinformatician, myself, if I'm working with some collaborators, we have qPCR experiment. Once they are done in the biotechnology side, they give us the data set, which is made of different samples, giving the experimental design, and list of genes, including some housekeeping genes, to look into the background as needed and to normalize. What we are hoping to get is CT values, okay? So pay attention again to what is CT value in this case.
TR curve. The defined signal intensity has to be exceeded until the product is abundant enough so that the fluorescence is detectable over background. The cycle number at which enough amplified product has been accumulated to exceed that threshold is referred to as the CT value. qPCR is mainly performed using one of these two assays. In the cyber-green assay, the fluorescent reporter is an intercalating dye that binds to double-stranded DNA. The Techman qPCR method works with probes specifically designed to bind the sequence of interest. Upon synthesis of the complementary DNA strand, the probe is cleaved, which results in a fluorescent signal. In the cyber-green qPCR method, one PCR cycle starts with the separation of the two DNA strands of the amplicon. During denaturation, the primers bind to their complementary parts, flanking the sequence of interest. The polymerase starts with the synthesis. The cyber-green dye binds the double-stranded DNA fragments, and the fluorescence is monitored after each cycle. In the Techman assay, short DNA probes are provided, which are attached to a fluorescent reporter at the 5' end. The reporter activity is suppressed by the 3' quencher as long as they are in near proximity. The strands separate in the denaturation step. After this, the primers can bind, and the Techman probes are designed to specifically bind in between the target sequence of the amplicon. The TAC DNA polymerase has a 5' exonuclease activity, which will shoo away the target-specific probe. Cleavage of the Techman probe separates the reporter from the quencher, which leads to the emission of a fluorescent signal that can be detected after each cycle. In both assays, the increase of fluorescence along product amplification generates a curve. During gene expression analysis, multiple genes can be compared. The number of cycles after which the signal intensity reaches the threshold is shown as the CT value. The presence of gene 1 is detected after 21 cycles, whereas the presence of gene 2 is detected in cycle 25. In general, low CT values go along with high DNA concentrations, and high CT values mean that signals are detected late, indicating low DNA concentration. Quantitative PCR finds applications in various fields. It can be used for diagnostics to detect infections with pathogens, and it is frequently used for gene expression analysis. For more scientific content, check out the videos on this channel and make sure to subscribe to stay updated. Thanks.
So yeah, I hope this video helps to bring a good summary of all what was discussed before, and even it highlights very nicely some of the concepts in a very, I hope, clear way. So the CT value, as the threshold is defined in our experiment, once we get the outcome scores, it refers to the number of cycles needed for the target DNA or RNA sequence to be amplified as we look at specific genes. How many number of cycles are needed? That is when we start looking into to say the number of cycles to say recognize the CT value. Okay, so analysis of qPCR data. So I'm gonna skim as well through some of the ideas here, some as well interesting ideas. So here we have two types of, sorry, we have one-step real-time PCR, as you see in the left side here. And here we have two-step real-time PCR. In one-step RT-qPCR, that's called RT-qPCR, sorry, it is, yeah. So this is here. If you remember the previous, not the previous video, the one before, there's reverse transcriptase qPCR, and there is real-time PCR, which we refer to it as qPCR. So we can combine both of them into one step, or we can run these two protocols separately. Start with RT-PCR and then go to qPCR. Okay, so in case they are in one step, both the reverse transcription and qPCR occur in the same tube. And after the RT step is completed, after the RT step is completed, the qPCR step immediately follows without opening the tube or transferring the reaction mixture. As you see here, there is opening and then there is a transfer to another a tube in this case. So there would be a set of differences in terms when to use which, depending on the flexibility of the experiments, the sensitivity and the application. So usually the choice between these two often depends on the specific experimental needs. And the top right here, what we see as some of the concepts is RNase H. So it is an enzyme. This is an enzyme here, okay, RNase H, that recognizes and degrades the RNA strand of RNA DNA duplexes, okay. So when there's a hybrid molecules where one strand is RNA, and as you see here, and the other is DNA, okay, this degradation might be necessary for efficient synthesis of double strands, okay, of double strands to come. And so RNase H can play a role into that. So this is one of the roles of RNase H, to degrade the RNA strand of when there's RNA DNA. And also, however, one thing to note is, in this case, if RNase H acts too quickly or aggressively on the RNA strand, in this case here, you see the cut here, in this RNA-DNA hybrid, the RNA can be degraded before the entire cDNA synthesis process is complete. This actually can result in truncated or incomplete cDNA. So this refers to the process of creating the cDNA and keeping an eye on, the role of RNase H during this process. So anyway, in summary, the role and desirability of RNase H activity varies based on the molecular biology application one is having in hand. And what we see here is we're looking into two issues. When the primer itself spans an exon-intron boundary, so a gene would have, as we know, the genomic DNA have exons and introns. When the primer spans into these boundaries, what's going to happen is it will not be able to bind properly to the genomic DNA. Okay, they will not be able to bind properly if they go to these edges. You see here, they span this area. Yet the cDNA, as we see below here, one of the advantages of cDNA, which is derived from mRNA, it contains only exonic. There is no introns in this part because they have been removed. So the primer will find a perfect match in this case. Okay, makes it easier. in this case. So, and again, as we see here, primers can flank in introns. And so if primers are designed such that they flank a long intron in the genomic DNA, this is also where we can see differences between cDNA, the complementary cDNA, and the genomic DNA. And see that during PCR, the time provided for extending the primers and copying the target is often based on the expected size of the target. So in the case of cDNA, it lacks the introns, no introns in the cDNA. It can be helpful to distinguish it from, in this case, for the cDNA, the sequence is going to be shorter, as we can see. We will end up with a shorter sequence. Okay. So by strategically designing primers around exon-intron boundaries, this is the idea here, or around long introns, for example, it can be assured that cDNA are selectively being amplified and not the genomic DNA, because eventually this might be in the same sample. You end up with these two samples, right? And you have cDNA and genomic DNA. One need to be careful in designing the primers to find the right target. Another type of technology, which is something related to PCR as well, it's what's called droplet digital PCR. It's a method, droplet digital PCR. It's a method used for precise and absolute quantification of the nucleic acids, okay? It goes through different steps where we go here with partitioning and then usually end up with many droplets. Okay, you see many droplets, okay? And then the PCR is performed next for amplification. Eventually, the number of positive and negative droplets, those who are having certain intensities would be positive, those with no intensities would be considered negative. So the amplification in terms of that specific step, each droplet undergoes PCR amplification. Each droplet here is gonna go through an amplification. And by the end of the PCR, again, we will have both positives and negative droplets. As you can see here, also the PCR amplification in this case. The counting of the positive and negatives is going to help in measuring the difference or doing the quantification. This is one application, for example. If we take the swab and then from the target, imagine we want to measure if SARS-CoV-2 is there or not, we can do this through two different ways, either by RNA extraction, which will follow and go with the traditional or typical qPCR that we looked into, or we can directly use the crude lysate. So RNA extraction have an extra steps that are needed to get as compared to the crude lysate in this case. Okay, and this material or the crude lysate can go directly to the droplet digital PCR. Eventually, it will help us to quantify again the availability of that mRNA target and so on and so forth. The crude lysate itself involved. It is just to refresh. It involves the lysis of cells or tissues, and so it is this material that is extracted from the cell itself, and it's a mixture obtained after breaking or opening cells or tissues. So again, as compared to going and do more steps to do RNA extraction, one can use crude lysate, and again depends on the specific needs of which technology to use. Let's look into this video here.
The human genome is of extraordinary complexity, with its 3 billions of DNA-based pairs that form the thousands of genes that make up our biology. Understanding this complexity requires powerful tools. Digital PCR is a powerful new tool for quantitative DNA measurements. Like real-time PCR, digital PCR uses a simple sample preparation that utilizes TaqMan primers and probes and an optimized super mix. Real-time PCR provides relative quantification. In contrast, digital PCR provides absolute quantification of target DNA sequences in the sample. Let's see how droplet digital PCR achieves precise DNA measurements. The key is sample partitioning. With droplet digital PCR, reaction mixes are partitioned into approximately 20,000 evenly sized droplets, each 1 nanoliter in volume. PCR is performed on each individual droplet. If a droplet contains the target sequence, it will be amplified and a TaqMan reporter dye will emit a fluorescence signal. If the target sequence is not present in a droplet, no PCR will take place and only weak residual fluorescence is emitted. By scoring each individual droplet for a fluorescence signal, droplet digital PCR achieves absolute quantification with a high level of precision and single molecule sensitivity. To generate digital results, samples are amplified to endpoint in a conventional thermal cycler. Droplets that contain the target DNA sequence are read as positive. Those that do not are read as negative. The fraction of positive droplets determines the concentration of the target DNA sequence in the sample. Now let's look at how the Bio-Rad QX100 droplet digital PCR system performs digital PCR with unrivaled precision and sensitivity. The QX100 system uses a simple workflow that requires minimum hands-on time and procedures that are easy to perform. First, 20 microliters each of 8 prepared samples are transferred to the droplet generation cartridge. Droplet generation oil is then loaded into the corresponding channels of the cartridge. There are 8 oil channels, 1 for each of the samples. Once the cartridge is loaded with prepared samples and oil, it is placed in the QX100 droplet generator. Using microfluidic technology, the droplet generator combines the samples and oil to create an emulsion of approximately 20,000 mono-sized droplets for each sample. Every droplet contains a nanoliter volume of the sample, including any target sequences that may be present. After the droplets are generated, the emulsified samples are transferred to a standard 96-well PCR plate, using a standard 8-channel pipette. The plate is then loaded onto a thermal cycler, where PCR is performed to amplify the target DNA to endpoint. When PCR is complete, the plate is loaded onto the QX100 droplet reader. The QX100 droplet reader is an automated liquid handling device equipped with optimized microfluidics designed for precise, reproducible detection of target DNA sequences, droplet by droplet. The droplet reader automatically sips each sample, singulates all the droplets from a sample, and then streams them in a single file past a 2-color fluorescence detector. The detector reads each droplet in turn at the rate of 1,000 droplets per second, and determines which droplets emit fluorescence and which do not. As each droplet is read, the data are recorded graphically by Quantisoft software. The software records the positive and negative droplets in each sample and plots the droplet by droplet fluorescence. This allows the data to be analyzed and visualized according to specific application and research requirements. The accuracy, sensitivity, and precision of droplet digital PCR enable a wide variety of applications that require accurate, absolute quantification. Examples of these applications include copy number variation, mutation detection, and gene expression. QX100 Droplet Digital PCR. The future of quantitative PCR is here, and it's digital. Be part of it.
So interesting. This certainly ends with a nice advertisement, but it's a very interesting technology that we are looking into. So we looked into this actually summarized some of the steps here. 1-step RT-DT PCR and 2-step QT PCR, and how there are different steps in terms of technology and library. Yeah. We're gonna look into this in the final part. We will skim through this and then talk about graphical abstract. Okay, so I'm gonna stop for here for now.